Inflammageing is defined as a chronic, systemic, low-grade, sterile inflammatory state that accelerates during the biological aging process. It is driven by endogenous danger signals rather than acute pathogens, primarily resulting from the reciprocal crosstalk between compromised mitochondrial networks, persistent cellular senescence, and the progressive decline of the immune system, known as immunosenescence. The biological aging process is fundamentally characterized by the development of chronic, low-grade, systemic sterile inflammation, a phenomenon biochemically termed inflammageing. Unlike acute inflammation, which represents a highly controlled physiological response to localized tissue injury or microbial infection designed to recruit immune effectors for pathogen clearance and tissue repair, inflammageing is a persistent state driven by endogenous stress signals. Successful aging, which is consistently observed in exceptionally long-lived cohorts and centenarians, correlates with significantly lower circulating levels of pro-inflammatory mediators. Conversely, a state of elevated inflammageing serves as a robust biomarker for biological aging, multimorbidity, and heightened mortality risk. It acts as a primary driver of age-associated degenerative pathologies, including cardiovascular disease, neurodegeneration, and metabolic syndromes. Historically, inflammageing was attributed primarily to a continuous lifetime antigenic load and cumulative environmental stress. However, modern molecular biology establishes that it is a far more complex phenomenon originating from the dysregulation of evolutionary conserved intracellular pathways. The primary endogenous sources of this sterile inflammatory state include the progressive accumulation of damaged macromolecules, the structural decay of cellular organelles, the presence of dysfunctional senescent cell populations, and the age-associated degradation of the immune system, structurally categorized as immunosenescence. At the cellular level, the initiation of inflammageing is deeply interconnected with cellular senescence. Cellular senescence is a state of permanent cell cycle arrest triggered by diverse forms of intra- and extracellular stressors, including severe DNA damage, telomere attrition, oncogenic activation, and profound oxidative stress. This arrest is mediated through the upregulation of cyclin-dependent kinase inhibitors, primarily the P16INK4A and P21CIP1/WAF1 pathways, which structurally block downstream cell cycle progression by keeping the retinoblastoma protein in a hypophosphorylated state. Although senescent cells lose their proliferative capacity, they remain highly metabolically active. Instead of undergoing programmed cell death via apoptotic pathways, these cells persist within tissue matrices and actively alter their microenvironment through the hypersecretion of a complex biochemical cocktail known as the senescence-associated secretory phenotype, or SASP. The persistent secretion of the SASP directly bridges localized cellular stress to systemic inflammageing, actively compromising surrounding healthy tissues. The composition of the senescence-associated secretory phenotype is highly heterogeneous and dependent on cell type and the specific senescent trigger. However, it consistently consists of pro-inflammatory cytokines, chemokines, growth factors, and extracellular matrix-degrading matrix metalloproteinases. Prominent canonical components include interleukin-6, interleukin-1 alpha, interleukin-1 beta, and tumor necrosis factor alpha. It also includes key chemokines such as interleukin-8 and monocyte chemoattractant protein 1. These secreted factors act via autocrine and paracrine signaling loops to reinforce the senescent state within the originating cell and actively induce senescence in adjacent, previously healthy cells, a process termed paracrine senescence. Furthermore, the continuous release of matrix metalloproteinases such as MMP-1, MMP-3, and MMP-10 actively degrades the structural scaffolding of the extracellular matrix. This breakdown disrupts tissue architecture and releases matrikines, which are bioactive extracellular matrix fragments that possess intrinsic pro-inflammatory signaling properties. This localized degradation creates a positive feedback loop that intensifies sterile inflammation. In a young organism, the acute induction of the SASP serves a physiological purpose, actively recruiting innate immune cells, such as macrophages and natural killer cells, to target, clear, and eliminate the senescent cells, thereby facilitating tissue remodeling and repair. However, during biological aging, this clearance mechanism fails due to immunosenescence. The progressive decline in the functional capacity of both the innate and adaptive immune systems compromises the surveillance and phagocytic clearance of senescent populations. Consequently, senescent cells accumulate exponentially within adipose tissue, skeletal muscle, the liver, and the vascular endothelium. This accumulation transitions a localized, transient repair signal into a persistent, chronic source of systemic, low-grade inflammation. This systemic state actively impairs localized stem cell niches, suppresses tissue regeneration capacity, and directly drives the clinical manifestations of multimorbidity. The underlying molecular trigger driving the hyperactivation of the SASB within senescent cells is intrinsically tied to severe mitochondrial dysfunction. Mitochondria undergo profound structural and functional alterations during aging and cellular senescence, an altered state often referred to as mitochondrial dysfunction-associated senescence, or MIDAS. These changes include a marked loss of mitochondrial membrane potential, structural swelling, disrupted Christie architecture, and a severe compromise in the efficiency of the electron transport chain complexes. Consequently, the coupling of the proton gradient to adenosine triphosphate synthesis is impaired, resulting in a dramatic reduction of cellular ADP production. To compensate for this bioenergetic deficiency, dysfunctional mitochondria shift toward glycolysis and exhibit an uncontrolled leakage of electrons from complexes 1 and 3 of the electron transport chain. This electron leakage results in the intracellular overproduction of reactive oxygen species, specifically superoxide anions and hydrogen peroxide. This sustained state of oxidative stress causes cumulative damage to vital intracellular components, inducing lipid peroxidation of membranes, the carbonylation of structural proteins, and the induction of double-stranded DNA breaks within both the nuclear and mitochondrial genomes. This continuous leakage of mitochondrial reactive oxygen species acts as a potent upstream, signaling mechanism that activates the nuclear factor kappa B, or NF-κB, transcriptional pathway. NF-κB is the principal transcription factor governing the expression of the majority of pro-inflammatory SASB genes. Concurrently, the accumulation of damaged, oxidized intracellular macromolecules acts as an endogenous danger signal that directly triggers the assembly and activation of the NLRP3 inflammasome complex. The NLRP3 inflammasome is a multi-protein intracellular platform comprising the sensor molecule NLRP3, the adapter protein ASC, and the effector protease caspase 1. Upon activation, caspase 1 proteolytically cleaves the inactive precursor proteins pro, interleukin 1, beta, and pro, interleukin 18 into their mature, highly potent, active pro-inflammatory forms. This processing directly accelerates the inflammatory cascade. Furthermore, mitochondrial dysfunction during senescence is characterized by a significant decrease in the NAD+ to NADH ratio. This metabolic shift suppresses the activity of sirtuins, specifically SIRT1 and SGRT3, which are NAD+ dependent deacetylases that normally inhibit NF-κB transcription and promote mitochondrial biogenesis. The loss of sirtuin activity removes the molecular brakes on the inflammatory response, allowing the NF-κB pathway to run completely unchecked, which exponentially amplifies the cellular production of IL-6 and TNF-alpha. The intracellular inflammatory environment is further intensified by the failure of mitochondrial quality control mechanisms, specifically mitophagy. Mitophagy is a specialized form of selective autophagy dedicated to the identification, isolation, and lysosomal degradation of damaged or depolarized mitochondria. In healthy cells, this process is strictly regulated by the PINK1 and Parkin signaling pathway. When mitochondrial membrane potential collapses, PINK1 accumulates on the outer mitochondrial membrane, recruiting the E3 ubiquitin ligase Parkin to phosphorylate and ubiquitinolate outer membrane proteins, signaling the autophagosome to engulf the damaged organelle. However, during cellular senescence, mitophagy pathways are heavily downregulated due to the transcriptional suppression of key autophagy reception genes such as BNIP3 and PINK1 itself. This downregulation results in the intracellular retention of highly damaged, fragmented, and uncoupled mitochondrial networks that escape degradation. These persistent, damaged organelles continue to generate high levels of oxidative stress while leaking metabolic intermediates into the cytosol. When these uncoupled mitochondrial networks undergo structural rupture due to the accumulation of membrane lipid peroxides, they release inner mitochondrial constituents directly into the evolutionary distinct environment of the cellular cytoplasm. This includes cardiolipin, mitochondrial transcription factor A, and, crucially, unmethylated mitochondrial DNA, or mtDNA. Because of its bacterial endosymbiotic evolutionary origins, mitochondrial DNA retains distinct structural features, such as unmethylated CpG motifs, that closely resemble bacterial DNA. Consequently, when it is displaced into the cytosol, it is recognized by the cell as an endogenous pathogen-associated molecular pattern, signaling an immediate intracellular threat. The presence of cytosolic mitochondrial DNA is bound and sensed by the intracellular enzyme cyclic GMP-AMP synthase, or cGAS. Upon binding to the sugar phosphate backbone of the displaced mtDNA, cGAS undergoes a structural conformational change that activates its catalytic domain. This domain catalyzes the synthesis of the secondary messenger cyclic GMP-AMP, or cGMP, from intracellular ADP and GTP. The generated cGMP molecules subsequently bind to and activate the stimulator of interferon genes, known as the STING receptor, which is localized on the endoplasmic reticulum membrane. Upon activation, the STING receptor undergoes oligomerization and translocates from the endoplasmic reticulum to the Golgi apparatus. This translocation induces the downstream recruitment, phosphorylation, and activation of tank-binding kinase 1, or TBK1, and the transcription factor interferon regulatory factor 3, or IRF3. This signaling cascade works in tandem with the canonical NF-κB pathway to drive the robust transcription and systemic secretion of type 1 interferons, specifically interferon beta, along with an array of additional pro-inflammatory chemokines. This specific molecular event establishes a continuous, self-sustaining intracellular inflammatory loop. The cGAS-STING pathway and the NLRP3 inflammasome effectively lock the senescent cell into a state of permanent hyperinflammation. This state continuously fuels the systemic SCSB cloud, maintaining the low-grade sterile inflammation characteristic of inflammageing. Furthermore, the cGAS-STING pathway actively reinforces the cell cycle arrest of the senescent cell by inducing p53 activation, preventing the cell from ever re-entering the cell cycle while ensuring it continues to secrete inflammatory mediators. This crosstalk between the nucleus, the decaying mitochondrial network, and the cytosolic innate immune sensors creates a chronic inflammatory feedback loop that cannot be resolved by conventional anti-inflammatory mechanisms. This profound realization has completely shifted the landscape of modern longevity research. We now understand that we cannot treat the diseases of aging individually. We cannot treat heart disease, cognitive decline, and joint degradation as separate issues when they all share the exact same root cause: the toxic crosstalk between failing mitochondria and cellular senescence. If we want to truly extend human health span, we have to intervene at the exact point where this inflammation cascade begins. Intervening in this specific pathway by therapeutically suppressing senescent cell survival mechanisms using senolytics like dasatinib and quercetin, or upregulating mitophagy via NAD+ repletion to restore mitochondrial quality control, represents the primary target in modern longevity medicine to halt the systemic cascade of biological aging. By clearing out the zombie cells and preserving mitochondrial membrane integrity, we can break the vicious cycle of cellular corruption, lowering systemic inflammaging and preserving tissue function down to the molecular level.